Good morning, and hello, everyone. I'm Sally Head-Dahlquist, President of Immunochemistry Technologies. And yes, that is a giant light bright I'm standing in front of at the 2017 AACR Cancer Research Meeting. Thanks for joining our webinar today. It's October and Halloween is almost here, so we thought you'd want to learn a little bit more about cell death. Today we're going to focus on apoptosis, pyroptosis, necrosis, and necroptosis, which really blurs the line as programmed necrosis, and touch on some of the others like autophagy and ferroptosis. Again, I'm Sally Dahlquist, and I'm a biochemist. I have a BS in genetics and cell biology and an MBA. I started out working in the lab, helping to develop assays for apoptosis, and now I'm president of the company. So Immunochemistry Technologies is a biotechnology company, and we're on a mission to help cure disease, and we do that, ironically, by studying cell death. We sell reagents to help researchers understand the unique biochemistry of each cell death pathway. Our customers want to know if the disease or the treatment is killing cells or keeping them alive, and they use our products to do that. And ultimately, this will help them discover better drugs, and that's why we're all here, to help cure disease and save lives. Assisting me today is Allison Westcott, our Customer Service Manager, so a big thanks to Allison for helping. And to those of you out there watching, we really appreciate you taking the time to make this webinar part of your day. We've muted your phone, but we really encourage you to ask questions, so just type them into the chat box or email them to help@immunochemistry.com, and we will respond directly to you after the webinar. And also, we're recording this, and we'll post it on our website and YouTube and email a link to you, so you'll be able to watch it again at any time and share it with your colleagues. And here's our agenda for today. We'll talk about cell death, go over the different types, and look at a couple of the kits from ICT. I hope this webinar will give you some inspiration when setting up your next experiment. Cell death is a vital part of life for all species, from plants to humans, to maintain homeostasis and prevent disease. Cell death is good for you when it's controlled, like leaves falling off the trees, and helps maintain our bodies to grow and remodel as we age. Cell death is bad when it's uncontrolled and leads to birth defects and diseases like cancer, where cells are growing out of control and they should be dying, or neurodegeneration and dementia, where cells are dying too soon and they should be living. It may also lead to autoimmune disorders. In diseases like a heart attack or stroke, there's a lot of tissue damage, and being able to assess the extent of damage can provide valuable diagnostic information. With cancer, being able to image if and when tumors respond to chemotherapy and start dying will lead to better care, enabling doctors to quickly select the most effective drug and minimize the bad side effects from ineffective treatments that don't actually kill the tumors. Cells die differently. Our bodies are very complex and do their best to keep us healthy. And some ways our cells do that is by responding to an accident. That's necrosis, immediate death that triggers an immune response to form a scab on a cut or repair the injury. And that immune response also triggers the cells around the injury or even a tumor to die as well. That causes necroptosis, which is programmed necrosis that releases other immune molecules. Sometimes our cells sacrifice themselves to save the body overall. Think of the cytokine storm of reaction. It may also trigger regulated cell death via apoptosis to dismantle damaged cells in ways that don't trigger inflammation. We'll talk mainly about these four pathways: necrosis, necroptosis, apoptosis, and pyroptosis, and touch upon a few of the other cell death pathways like Oncosis, ferroptosis, and autophagy that are interlinked with each other. Each one of these biological pathways has a unique biochemistry and pathophysiological characteristics, and it's important to understand all the biochemicals involved so we can develop better treatments and drugs with fewer side effects. It's an amazing process that fascinates me. So let's dig into the science behind cell death. Cell death is typically categorized into non-programmed or programmed cell death pathways. The non-programmed cell death is accidental cell death, or ACD, and necrosis is what we typically think of a dead cell. It is usually due to an injury, accidental death, or the body's inability to repair itself. That's what we see on the left with our zombie scientists. When the cell is immediately injured, it goes into oncosis and swells. Oncosis is the passive, pre-lethal phase of death, leading to necrosis when the cell simply breaks down, spills its contents into the surrounding tissues, and damages them too, which snowballs and creates more damage if the body doesn't heal itself by launching an immune response. On the right, we have programmed or regulated cell death that has an underlying genetic program. As the cell ages, it follows a genetic pattern for growth, maturation, and replacement. Programmed cell death is what happens when the cell gets too old or becomes dysfunctional that we see on the right. The cell efficiently dismantles itself and is recycled without damage to the surrounding cells. Scientists used to think that there was just one form of programming called apoptosis, like the leaves falling off a tree. But now we know there are several other pathways of programmed cell death that trigger a biochemical response to control the breakdown of old, damaged, or dying cells, like necroptosis, and can lead to inflammation. And that's where it gets really interesting. There are many biochemical pathways guiding cell death, and researchers are classifying them in different ways. This is figure 1 from Jan's 2019 paper on cell death modalities. Thanks to their lab at the Johannes Gutenberg University in Germany for all their great work. Researchers look at the initiation of the death signals. What stimulated the cell to die in the first place? Was it an injury? That's necrosis, which is accidental cell death on the left in orange. And when the necrotic cell bursts open, that triggers an immune reaction that leads to some forms of programmed cell death like necroptosis. If it wasn't an accident and the cell was programmed to die, what biochemical told the cell to die? If it were the caspase cascade, it would be apoptosis, which you see in light blue in the lower right. If it were not apoptotic cell death, what else was happening? Was it aging or an infection, an internal or an external signal? For example, pyroptotic cell death is triggered by an infection, which is an immune reaction launched by caspase-1. We also look at cellular morphological characteristics like vacuoles and autophagy and the molecular mechanism, like iron ions involved in pyroptosis. This is also from Jan's 2019 paper based on the classification. And here's a great sketch of the morphology of the cells, looking at cell size, the membrane integrity, chromatin density, organelle arrangement, and the presence of vacuoles. Accidental death, necrosis, is in the upper left, next to apoptosis, which is programmed cell death. Everything else on the slide is non-apoptotic programmed cell death under the dark blue banner. However, these pathways are not isolated from each other. There are many biochemical overlaps. So necrosis is associated with membrane rupture and release of intracellular inflammatory factors that can give rise to inflammation, as seen in necrosis, necroptosis, netosis, and pyroptosis. In comparison, apoptotic cells do not stimulate inflammation as they are rapidly cleared by phagocytes. But if they're not cleared, they can become necrotic. Mitosis can turn into autophagy or apoptosis. The morphology is similar for many of the classifications, and notice that some cells are rupturing. The cell membrane is compromised, like necrosis and necroptosis, of course, and pyroptosis and netosis are immune reactions, and parthenatose for mitochondria. Some form vacuoles during cell death, like autophagy, which is our intracellular degradation system, and there's entosis, methuosis, and pyroptosis. In some, the DNA fragments, like apoptosis and pyroptosis. So now let's look at some live cells. I love these pictures because you can actually see what the cells look like. This is from a scanning electron microscope taken by Xin Chen and researchers from Xiamen University in Fujian, China. The normal cell is on top compared with the others, which are regulated cell death pathways. In necroptosis on the left, you can see the swollen cell is about to burst due to osmotic swelling from ion influx. Necroptosis is mediated by the death receptors TNFR1, TRAIL, and FAS, and activated by receptor interacting proteins RIP1 and 3. Necroptotic cells assemble the necrosome, which is a multi-protein complex resembling amyloid. And once that cell bursts, it will release DAMPs, which are damage-associated molecular patterns, causing an inflammatory reaction. In apoptosis, in the middle, caspases are activated. The cell condenses, and the cellular contents are neatly packaged into balls, the apoptotic bodies that are ultimately taken up by phagocytic cells in a non-inflammatory manner. The plasma membrane does not rupture. Instead, it's engulfed by phagocytic cells and does not cause an immune reaction. Apoptosis requires energy. It is ATP dependent, and if an apoptotic cells run out of ATP, it will collapse, resulting in secondary necrosis, so it swells and lyses. Compromised apoptosis can also shift to necroptosis, and necroptosis can shift into apoptosis. The suppression of apoptosis by inhibiting caspase enzymes reveals necroptotic pathways as if a backup system for cell death. The activation of tumor necrosis factor receptor, TNFR, can stimulate both apoptosis and necroptosis, showing their interactive processes. By comparison, on the right, the inflammasome is formed in pyroptotic cells, which is a multi-protein complex that contains caspase-1 and is triggered by NLR activity. The pyroptotic cell flattens and forms blebs of pyroptotic bodies. The DNA fragments, but the nucleus remains intact, which is different than apoptosis. The plasma membrane will rupture and release the intracellular contents, resulting in inflammation. In both necroptotic and pyroptotic cells, the plasma membrane ruptures, but it happens differently. It's the pore-forming activity of their biochemistry that makes the difference. In necroptosis, MLKL, which is mixed lineage kinase domain-like oligomers, form selective channels that cause an influx of ions, whereas in pyroptosis, it's Gasdermin D that forms non-selective channels after its cleavage by caspase-1 or 11. Accidental death by necrosis is inherently inflammatory. It's not shown here. And that may also trigger a regulated pathway such as necroptosis or apoptosis in surrounding cells. For example, oncosis leading to necrosis is frequently seen inside the body from oxygen deprivation events like ischemia. And necrosis is often found inside the core of a tumor due to the hypoxic microenvironments, while the outer penumbra region tends to contain apoptotic cells. The type of cell death in surrounding tissues can also affect tumorigenesis. The tissue microenvironment found around cells undergoing necroptosis directs the lineage commitment in liver cancer. There are many molecules involved in these different cell death pathways. One common thread to both apoptotic and pyroptotic cell death is the activity of caspases. Caspases are enzymes underlying the cell death process. Caspases, or cysteine-dependent aspartate-directed proteases, are activated and then cleave substrates, leading to the eventual disassembly of the cell. There are 4 different groups of caspases, those involved in apoptosis and those involved in inflammation and keratinization. In terms of apoptosis, initiator caspases, which are 2, 8, 9, and 10, regulate apoptosis upstream by initiating caspase cascades, while the effector caspases 3, 6, and 7 are responsible for proteolytic cleavages that lead to cell disassembly. So they are the ones that take apart the cell. In terms of inflammation, that is heavily dependent on the activity of caspase-1, which is associated with inflammasome activity like the NLRP3 and is a key housekeeping enzyme in its conversion of pro-interleukin-1 beta protein into the active interleukin-1 beta cytokine. Caspase-1 activity is integral to pyroptosis. So caspases can be activated by external or internal signals, and extrinsically, they're activated by death receptors. There are several death receptors that can activate apoptosis shown in the table. There is also an overlap with these processes like the death receptor tumor necrosis factor receptor 1. TNF is the death ligand on the cell that you see on top. And the activation of TNFR1 can lead to cell survival, apoptosis, or necroptosis, depending on the biochemistry involving RIP1 and ubiquin E3 ligases. It can generate the pro-survival complex 1 and LIV, which you see on the left. But if it activates caspase-8, the cell enters apoptosis in the middle and is dismantled without inflammation. If caspase-8 is inhibited, which you see on the right, it assembles the necrosome, that multi-protein complex, that leads to MLKL oligomerization and membrane permeabilization that creates selective ion channels. This causes the characteristic osmotic swelling and bursting that leads to inflammation. This is a nice table from 2019 created by Mary Choi of Weill Cornell Medicine in New York. It highlights some of the features and key regulatory molecules involved in some of the cell death pathways. As expected, only necrosis does not have any key molecules. The rest have key proteins like caspases or iron or MLKL to regulate the pathway. Notice that only 2 of the pathways are not genetically regulated, and that's necrosis and senescence. Senescence is the cell cycle arrest, typically linked to aging. So remember, we are recording this, so you'll be able to come back and look at this slide at more time. Now that you know what the cells look like, let's review each pathway. Oncosis is the pre-lethal phase after an injury leading to necrosis. This is caused by the disruption of ionic pumps such as calcium influx. The cell swells and permeabilizes. There's also karyolysis, vacuolation, and this is a passive form of cell death due to the lethal cell injury. The cell loses injury and the ion pumps fail, leading to leakage and damage to the surrounding cells, which is inflammatory. I think of oncosis as the swelling and necrosis as when the cell ruptures. Oncosis is frequently observed in ischemia and could act as a link between autophagy, apoptosis, and necrosis. Necrosis is a form of cell death that does not involve activation of programmed cell death pathway to control the breakdown of the cell. It's considered a passive process and is stimulated by external factors like an infection, toxins, or an injury. These injuries lead to physical damage like cytoplasmic swelling, that's oncosis, rupture of the plasma membrane, loss of the organelles. And necrosis is a lytic form of accidental cell death that results in the release of DAMPs. Those are the damage-associated molecular patterns and propagation of inflammation. Overall, at its most basic level, necrosis is the sum of the changes that have occurred in the cells after they've died, regardless of the pre-lethal process. Necrosis, therefore, refers to the morphological state seen after a cell has died and reached equilibrium with its surroundings. Necrosis is easily detectable with several DNA binding dyes like PI and 70AD. Necroptosis blurs the line between necrosis and regulated cell death. The cell may look like necrosis and is morphologically similar with swelling and rupture, but necroptosis is regulated by underlying genetic programs and regulated by distinct proteins, mainly RIPK1 and 3. Necroptosis forms ion channels that cause the cell to swell and explode. Cell surface receptors, death receptors, toll-like receptors, and the T cell receptor activate RIPK. It is activated by RIPK1 and requires RIPK3 dependent phosphorylation of the MLKL. That MLKL oligomerization results in that plasma membrane rupture, leading to a lytic form of cell death, again associated with the release of the DAMPs. Both RIPK3 and MLKL are key effectors of injury propagation, and necroptosis represents a form of regulated cell death that's implicated in many diseases like kidney and lung injury. But on the other hand, necroptosis may have a beneficial role by removing infected cells by helping with the modulation of host defense. It may also activate anti-tumor response and help treat cancer. When researchers think of programmed cell death, apoptosis is often the first method that comes to mind. This isn't surprising, as apoptosis was the first well-characterized program of cell death and is the most widely recognized. The apoptotic pathway can be activated 2 ways: intrinsically by intracellular signals from inside the cell, or extrinsically from external signals like the death receptor. The intrinsic pathway is activated following internal cellular stress, which results in mitochondrial perturbation and the release of cytochrome C. Once released, cytochrome C binds to apoptotic protease activating factor 1, triggering the formation of the apoptosome complex, resulting in the activation of initiator caspase 9. An activated initiator caspase 9 then processes and activates effector caspases 3, 6, and 7. The extrinsic pathway is initiated following the activation of death receptors of the TNF superfamily. The trimerized receptor ligand complex recruits procaspase 8 to the death-induced signaling complex via the FAS-associated death domain, that's FADD, leading to the rapid activation of caspase 8. Activated initiator caspase 8 then processes and activates effector caspases 3, 6, and 7, which are responsible for the phenotypic changes associated with apoptosis. So once activated, the nucleus condenses, the DNA is damaged, the contents of the cell are neatly packaged into apoptotic bodies, and the cell is phagocytosed, meaning it is carried away without any injury to the surrounding cells, nor is there any inflammation. ICT has many products that can detect caspase enzymes, so researchers can easily track dead and dying cells in vitro, in vivo, and in real time, and I'll show you some data in a moment. Pyroptosis is a highly inflammatory form of programmed cell death that occurs upon infection and likely plays a role in the host's antimicrobial response to pathogens. Inflammation sensors like NOD-like receptors, which are NLRs, of infected macrophages recognize the flagellin components of pathogens and initiate the formation of multi-protein complex inflammasomes, which activate caspase 1, which then processes and activates interleukin-1 beta and interleukin-18, which are secreted from the cell. Gasdermin D and E are cleaved, leading to pore formation and swelling. The DNA fragments, but the nucleus remains intact, which is different from apoptosis. And also, caspase 11 can be directly activated by bacterial lipopolysaccharide, which is LPS, and induce pyroptosis. In a 2020 paper by Yuan Fang from Nanjing Medical University in China, there are now 3 proposed pathways of pyroptosis. There's the canonical and the non-canonical inflammasome pathways, and then in C, there's a newfound 3rd pathway that activates caspase 3. How can you detect cell death? Well, easily with detection kits from ICT. I'll mention just a few of them today. One of them is our Apoptosis versus Necrosis Kit. It contains 2 reagents. FLICA measures the caspase positive cells in green, and 7A-AD is a red fluorescing live dead stain that enters cells with compromised membranes and binds to DNA. It will not enter living cells with intact membranes. And so the slide shows images of your cat cells treated with sterosporine for 4 hours. And here you can easily see early apoptotic cells staining only in green with our FLICA caspase reagent, and cells in late apoptosis stain green and red, and necrotic or dead cells stain red only. Let's see with the arrows. This kit is ideal for monitoring cells by flow cytometry. So when you plot the FL1 channel by the FL3 channel, you can easily identify these 4 cell populations as they show up in separate quadrants. The lower left quadrant will house the live unstained cells. The lower right quadrant will contain the green staining early apoptotic cells. The upper right section will contain the green and red late apoptotic cells. And the upper left section will house the dead cells that have stained red. When we compare mock-treated and sterosporine-treated Jurkat cells that have been duly stained with our FAM FLICA green reagent and red 7A-AD, we see that the majority of the mock-treated cells fall into the live unstained category, while very few of the induced cells remain in this category. 46% of the sterosporine-treated cells are early apoptotic, and 50% are late apoptotic. We have many FLICA kits available that measure apoptosis and different caspases. FLICA is an inhibitor that binds to active caspases that you just add to the cell culture media. The reagent lasts through many rounds of processing and dual staining studies and are compatible with other stains like GSP, DAPI, hooks, DTMR, antibodies, morpholinos, etc. And you can analyze your data with a plate reader, flow cytometer, or microscope. These are cultured primary rat hippocampal neurons that were triple stained to determine if the cells are viable. By using the blue DNA stain hooks, we see there are 4 cells in A. By staining with the green FAM-FLICA, we can tell that 3 of the cells are apoptotic because they're green. By using propydium iodide, which is red, we can tell that 2 of the cells have been permeabilized, and they are red in C. In this way, we see 3 populations of the cells in D, which is a composite image. There is only 1 live healthy cell that is only blue. 1 cell on the bottom is in early apoptosis, it's blue and green, and 2 cells are in late apoptosis and becoming necrotic. They're red, green, and blue, meaning that they are becoming membrane compromised and are in the late stages of apoptosis rather than necrosis. If a cell were necrotic but not apoptotic, it would only be red with PI, as seen in C. But once we do the overlay, we can tell they're in late apoptosis. Cells labeled with FLICA can be counterstained with reagents such as the red live dead stains, propydium iodide, and 7-AAD, and nuclear morphology can be concurrently observed using Hooks or DAPI, a blue DNA binding dye. To detect pyroptosis, ICT offers 2 Pyroptosis and caspase-1 assay kits to detect caspase-1 activation in cell cultures, also using our popular FLICA technology. The kit is available in green or far red fluorescence and can be analyzed with a fluorescence microscope, plate reader, and flow cytometer. Nigericin is included in these kits as a positive control for pyroptosis studies. This reagent is a potent microbial toxin derived from Streptomyces hygroscopicus. It acts as a potassium ionoform, inducing a net decrease in intracellular levels of potassium, which is crucial for oligomerization of the NLRP3 inflammasome and activation of caspase-1. Nigericin requires signaling through the pannexin-1 to induce caspase-1 activation and interleukin-1 beta processing and release. It's been shown to generate a robust caspase-1 activation response in various cell lines, including JRCAT and THP1 cells. Human colorectal adenocarcinoma cells were grown in polarized monolayers and infected with wild-type Salmonella constitutively expressing M. cherry, which are the red cells. After 9 hours, the live cells were incubated with ICT's active caspase-1 reagent, FAM-YVAD-FMK, which is the green cell below, for 1 hour in growth medium, washed and fixed. The confocal image reveals an extruding cell that is infected, and many red M. cherry labeled Salmonella are visible. The infected cell is undergoing pyroptosis, as evidenced by the positive staining for active caspase-1, visible as increased green fluorescence compared to background levels of fluorescence in the surrounding caspase-1 negative cell. ICT's caspase-1 inhibitor reagent, FAM-YVAD-FMK, was used to monitor caspase-1 induction response in JRCAT cells treated with nigericin for various periods of time. A common cell pool was spiked with the reagent and divided into separate treatment groups, and starting with 24-hour samples, working backwards, 10 micromolar of nigericin was added to the cells, and the samples were incubated at 37 degrees throughout the induction process. Following their respective treatment exposure periods, the cells were washed and analyzed on an Accuray C6 flow cytometer. The amount of caspase-1 activity detected directly correlated to the duration of the exposure period. The longer the cells were exposed to nigericin, the larger the proportion of caspase-1 positive cells are found in the sample. The kits also work with a fluorescence plate reader, and in this case, these are THP1 cells that were plated in 12-well tissue culture plates and treated either with a negative control or PMA, which induces pyroptosis to cause differentiation into macrophages. So after 48 hours, the PMA-containing culture medium was removed. The wells were rinsed to remove any non-adherent cells, and fresh culture medium containing LPS was added to the induced sample wells. After a 2-hour exposure period, LPS-containing medium was removed. Cells were rinsed with PBS and trypsinized to remove adherent cells. The trypsinized cells were transferred to tubes containing SBS to inactivate trypsin, and cells were gently pelleted by centrifugation. The supernatant were carefully removed and samples were resuspended in PBS. Samples were then stained with FAM-YVAD, the caspase-1 reagent, for 1 hour at 37°C. Then the samples were washed 3 times and read on a fluorescence plate reader at 488 excitation and 530 emission using a 515 nanometer cutoff filter. In the pyroptotic induced population, the relative fluorescence units of the green fluorescent signal was nearly 2 times greater than the RFU of the negative non-induced population on the left. ICT also has an autography assay, which is in red, and that contains a proprietary cell-permeant aliphatic molecule that fluoresces brightly when inserted into the membranes of autophagosomes and autophagolysosomes. Our kit allows researchers to easily monitor autography using a flow cytometer equipped with a green-yellow laser and the appropriate filter. And the optimal excitation and emission is 590 and 620. ICT also offers other kits to detect mitochondrial membrane potential. These are orangey-red fluorescent probes, and they fluoresce inside healthy, intact mitochondria, but don't fluoresce when dispersed in the cytosol. ICT has many fast, easy, and reliable methods that can be used to evaluate cell viability and functionality studies in vitro, in vivo, and ex vivo. Which reagent is best for your experiment all depends on which instrument you are going to use for analysis and what other stains you're going to use with it. All of these kits are available online, so check them out and expand your research toolbox by including ICT's reagents for cell death. I hope this webinar was helpful today. This wraps up our presentation for today. If you have any questions, please feel free to send them through the chat box or email to help@immunochemistry.com, and we will respond directly to you. Of course, you can always call us. We love hearing how you are using the products and helping you set up your experiments. So again, thanks for tuning in today and for all the research you're doing. And thanks again to Allison Westcott, Nick Wenninger, and Kristen Pauley, our marketing team, for managing these webinars. We here at ICT are proud to be part of the medical research community and provide research reagents and kits that can help understand and someday cure disease. I'm Sally Dahlquist, President of Immunochemistry Technologies, and it's been a pleasure helping you understand cell death. Have a great day.